We're all part of the Population Biology Behavioral Ecology group led by Dr. Greg Okoye-Crow. He couldn't be here today, so my name is Heidi Pagan, and this is Sarah Rogers and Tatiana Ferrer. We're all going to be presenting updates on our projects. A little bit of background on our role in the dolphin research. We are lucky to be part of Harbor Branch, who has this long-term study on the dolphins, and as you've already seen, there have been years of data collected already on the unusual mortality event just recently, and then years of strandings and disease and general dolphin health. So our goal is to come in and incorporate a genetics aspect to this research. And this is our website for our group. I'm going to go ahead and start by handing this over to Sarah. We are going to talk about a 3-part investigation, and she's going to be starting it off.
Thank you, Heidi. All right, so my name's Sarah Rogers again. I'm going to talk today about the population structure and dispersal of the dolphins along the east coast of Florida. So quickly, bottlenose dolphins, they're a very common marine mammal. They're found worldwide. They live in these interchanging communities. They have sex-specific associations, and they're apex predators. They've also been characterized, as you heard before, as sentinel species, which means changes in their environment may alter their physiology or behavior, which serves as an early warning signal to potential risks of their ecosystem and anything of lower trophic levels. Now, bottlenose dolphins along the east coast of Florida are managed in groups called stocks, and these are groups of marine mammals of the same species who share a common spatial arrangement and who interbreed when they're mature. There are 4 main estuarine stocks and 2 offshore stocks of the east coast of Florida. Now, dolphin studies have reported that there's an estimated around 1,300 animals within the Inner River Lagoon, but this is unpublished. And it's considered one stock all the way from New Smyrna Beach and Mosquito Lagoon down to Jupiter Inlet. There's 3 to 5 communities within the IRL based on whether you look at ranging patterns of dolphins or social affiliations. The IRL is a separate to an adjacent coastal population of dolphins that has been observed having limited to no movement between the two, essentially making the IRL dolphins a closed population. Finally, the population structure published to date has used limited number of samples or non-traditional methods, leading to misinterpretations of results. Now, you heard before about all these problems for dolphins. Adam mentioned the prevalence of infectious diseases, the pollutants. You heard about unusual mortality events with Steve, the interactions with crab pots and boats, and human encroachment and competition for their prey. So it's of extreme importance that we resolve the patterns of the dispersal, gene flow, population structure, and immune response in IRL dolphins. We first need to know who these animals are and where they go so we can then determine who specifically is actually at risk. Only then can we start to minimize the negative impacts dolphins face within the IRL. Now, there's 2 main ways to identify structure. One is observational studies, such as photo ID and radio tracking, but as Heidi mentioned, observation studies can only tell us so much. The second way to identify population structure is using genetics. For more concrete answers, genetic analysis using a multitude of markers is needed to understand what really may be going on within and among populations. For this study, we used 2 types of DNA, mitochondrial DNA, seen in red, and nuclear DNA, seen here in blue. Mitochondrial DNA, or mtDNA, is found in the mitochondria. It's maternally inherited, meaning you only get it from your mother. It's relatively high in mutation rate, but it only provides information to us on female patterns of dispersal. Nuclear DNA, specifically I use these regions called short tandem repeats called microsatellites that you actually heard from Josh as well. These are found in the nucleus. They're biparentally inherited, they're genetically hypervariable, and they offer insights on breeding patterns and male-mediated gene flow. Now, this study used over 600 samples that were collected over 10 years from New Smyrna Beach all the way down to Jupiter, inside and outside of the lagoon along the Atlantic. Now, skin was taken from dead or live animals and was immediately preserved or frozen. Once in the lab, the skin samples were extracted using a standard salt method, and DNA was quantified and archived. We then amplified mitochondrial DNA using standard primers, and an additional 10 microsatellite markers were used to amplify their respective regions. Sequencing on both strands of the mitochondrial DNA and genotyping of the nuclear DNA were done in-house here at Harbor Branch. as well as the alignment and analysis of all the data that was generated. Again, from start to finish, nothing in our lab gets sent out. Everything is done in-house to ensure optimal quality control and proper interpretation of the results that we find. So what questions can we ask with all of these samples covering such a large area over all these years? Well, we decided to take a 4 ecological avenue approach. We first started with an ecotype question, which is in the form of comparing dolphins compared within 2 ecosystems, the estuarine and the coastal dolphins. Our second hypothesis was based on subdivision of water bodies, or separate embayments, mosquito lagoons separate from the IRL proper versus the Atlantic. Third was a habitat-based approach, specifically using discrete hydrologic and geographic segments as potential boundaries. Again, you've seen this earlier on the segment maps. Fourth, we employed a more biological-based approach using the results from one of our collaborative studies Adam mentioned, using social networking and association patterns of IRL dolphins. Here we tested those population structure behind the 6 communities. So what did we find? We found that dolphins sampled from the IRL estuarine system are genetically differentiated from animals sampled along the Atlantic coast. Our markers showed a difference in the sexes, that males moving from both populations versus our IRL females, which seem to remain homebodies. We also were able to determine these differences have been in effect for a long period of time. And finally, with our extensively large sample size spanning the entire Indian River Lagoon estuarine system and all along the Atlantic, we were able to detect immigrants in both source populations seen here. In hypothesis 2, when we looked at water bodies, we found mosquito lagoon dolphins were actually more genetically similar to Atlantic dolphins than to IRL proper dolphins. Even more surprising was that these dolphins weren't a separate population. They're what's called a mixed stock, and that appears to have a minimal breeding between those 2 source species. Very interesting ecological situation that we're definitely looking at right now. When we tested the hydrologic and geographic segments that have been used in many previous dolphin papers as potential groupings, we found that there was minimal to no differences between these groups, and therefore, our segment map actually looks more like this, and we conclude that IRL dolphins aren't actually using the water management districts as boundaries to separate themselves genetically. Finally, using a biological boundary estimator of dolphin communities, we found, similar to the segments hypothesis, that dolphins sampled living within each of these community boundaries do not appear to be kin-based. And again, our results showed the fluidity of genes within the lagoon. So all this work helps us how, you may ask? Well, you have to start with a solid concrete foundation to build bigger and better projects upon. These findings will be the basis of all future dolphin-related studies, and we are very, very thankful for the PLATE supporters that have helped us finally nail down these answers. Further, your donation funds our continuously growing tissue and DNA archive, which we bank samples for future studies with collaborators all around the world. So now we can identify who actually is an IRL dolphin, regardless of where they may be found, and how these 2 populations overlap. So we can ask major questions like, do dolphins with IRL genes have different immune potential than Atlantic dolphins? Could those male dolphins be the disease vectors between the IRL and the Atlantic dolphin populations? Could dolphins with Atlantic genes be more adept to deal with pollutants, metals, and other toxic contaminants? Could IRL dolphins have a stronger immunity during those mass die-offs, those unusual mortality events? Is that why we haven't seen the mass mortality within the lagoon that we've seen during the coast? And what if we were able to determine which populations may be more at risk for these types of events ahead of time? Now, answers to these questions and more may be in the genes involved in the immune response, which, lucky for you, Tatiana will now tell you about in part 2 of our investigation.
Thanks, Sarah. So first I'm going to talk to you guys about why diversity in MHC loci matter. So understanding the genetic aspects of the immune response provides insight to the determinants of individual fitness and population viability. Immunocompetence is an individual's ability to produce a normal immune response post-antigenic exposure. And pathogens are some of the strongest selective forces, and individuals are constantly exposed to them, especially in changing environments. And low genetic diversity is associated with decreased fitness and increased risk of extinction.
So the major histocompatibility complex, or MHC, is the immunologically most important family of genes determining a population's ability to detect and combat pathogens. It is comprised of about 4 million base pairs and contains the most variable functional genes described in vertebrates. So the MHC is central to the vertebrate immune system. It is a system of recognition and presentation that initiates the immune cascade. It responds to pathogens by recognizing non-self peptides, and it determines the disease susceptibility or resistance at the genetic level. Here we have a basic schematic depicting a pathogen being engulfed by a macrophage, broken into pieces and presented by a receptor on the surface of the cell. A T cell then binds, activates, and turns on the cell, initiating the immune cascade. For this project, we are focusing on the binding and presenting that initiates the immune response in the individual, seen here in pink. So talking about our specific locus, the DQ is a heterodimer comprised of an alpha and a beta chain. DQ genes play prominent roles in adaptive immunity. Many associations have been made between the polymorphism seen in DQ genes and resistance or susceptibility to a broad variety of infectious or autoimmune diseases. Information on non-model organisms is lacking. Wild or semi-wild animals, like our bottlenose dolphins, are considered to be better models for studying adaptive survival under pathogenic conditions. And finally, DQ genes will hopefully provide insight on how different environments can influence MHC evolution in marine mammals. Of the DQ genes, DQA is one of the least studied in cetaceans. We targeted exon 2 for this study because it codes for the peptide binding region, which is responsible for antigen recognition, binding, and presentation to the T cell receptor, which initiates the immune response in vertebrates. So what did we find? First, we were able to isolate and sequence the entire DQA exon 2. To date, this has never been done for bottlenose dolphins. Then we determined that it is possible to genotype individuals rapidly with direct sequencing. We also found diversity at this locus. We cloned their PCR products to verify that the alleles were legitimate and then to determine the allelic composition of a heterozygous individual. We detected patterns of geographic variation in allelic diversity, finding very different allele profiles for IRL and Atlantic dolphins. We have evidence of positive selection, indicating that this variation is happening despite genetic drift and gene flow that Sarah just told you about. And finally, we identified variation in the peptide binding pockets. So paving the way for future work, now that we have been able to characterize diversity in exon 2 for bottlenose dolphins, and we've identified crucial variation in the pockets of the peptide binding region, we need to remember that this is just a part of the DQA gene. So some of the questions we asked ourselves during this project were, how does variation in the rest of the gene compare to exon 2? How can we look at the whole gene given the limitations of Sanger sequencing? And finally, how does variation in another immune response gene compare to DQA? Well, with that, we'll let Dr. Heidi Began tell you about part 3.
All right, thank you, Tatiana. So, like she said, I'm doing this last part. I couldn't talk about my project first without going over theirs because they laid the groundwork. Sarah just determined the population structure, and then my project is an extension of Tatiana's project where I'm looking at 2 different genes. So here's the gene that she was talking about, DQA, and just for a brief refresher, this gene has 5 exons and it has a promoter region at the very beginning. Transcription factors have to come and bind to that region before the gene can be turned on. When it's turned on, it's transcribed into an RNA molecule, which is translated into a protein. And then that protein is further processed by going through folding to become a specific conformation that allows it to form part of the DQ receptor molecule. Now it takes 2 genes to completely form that DQ receptor. She just told you about the importance of exon 2 in forming the peptide binding region. So I wanted to look at variation of this in both genes because that'll tell us about what types of different pathogens can be bound by this DQ receptor. But I also wanted to look at variation in the promoter region because that's going to tell us about how this gene could be turned on or off. So there are 4 different loci that I was looking at here: the DQA promoter, DQA exon 2, DQB promoter, and DQB exon 2. The next part of my project was to incorporate next-generation sequencing methods. These are high throughput methods. That means we can cover a lot more samples in a shorter amount of time and for a lower cost. The trick is that all the samples have to be combined into a single tube before they're loaded into the sequencer. So when we're amplifying our region of interest, we put on unique barcode pairs. That's a known segment of DNA at the beginning and the end of each region we were interested in. So each dolphin ends up having 4 unique barcode pairs. We did that for 95 dolphins and then combined all of those into a single tube. And that sample was loaded onto a chip in the IonTorrent personal genome machine and sequenced. We got more than 5 million sequence reads from this. And here's a little snippet of what the data looks like. It's not just the DNA sequence, it includes a bunch of symbols below that, which are quality values, which tell us how confident we are in each position of the sequence. So we did some filtering steps, and we ended up being left with more than 2 million sequence reads that were the correct length and had the identifiable barcode at the beginning and end of the sequence. Here's an example of the data processing. If these were our sequence reads, we'd sort them by the barcode. So here's a subset that had the same barcode at beginning and end of the sequences. We would then trim off the barcodes and align it to a reference sequence, and then we do our variant analysis. This is where, I'm sorry if you can't see very well, but there are green positions that are different from our reference sequence. So that's the parts that we're interested in. And here's an example of this for DQA exon 2 for one dolphin. It had over 1,000 sequence reads, and the positions that are a little bit lighter are the ones that are different from the reference sequence. So this was a nice sample because they fell very cleanly into 2 distinct groups. That's because we have 2 copies of the gene, 2 alleles, one from mom, one from dad. But most of our data was not actually that nice, so here's an example where there's a lot of noise. There are sequencing error and other amplification errors that we have to consider. So we had to run a bunch of controls as well. And I'm analyzing the data of the 2 different programs to see how well they line up. And then I'm ground truthing this because I include some of the same dolphins that Tatiana ran. And I can look at her data and mine and make sure they match up for those individuals. And that way I can confirm that this is a good method for using because she's doing traditional and then I'm doing next generation. So I'm still finishing up this analysis. When I'm done with that, this will be the most comprehensive MHC genotyping project for any cetacean. That was a lot to throw at you, so I'm going to do a quick summary of the 3 projects. The first one, presented by Sarah, was population structure and dispersal using non-coding loci. She was able to detect a genetic profile between estuarine and coastal dolphins and immigrants of each. She was able to resolve population boundaries and movements of Indian River Lagoon and adjacent Atlantic dolphins, essential to proper management and the basis of all future Indian River Lagoon dolphin research. Then Tatiana's project was differentiation using an immune-related locus. These are the first time this type of research has ever been documented for Tursiops truncatus. Provides us with a blueprint for the geographic variation and crucial binding pockets of the Indian River Lagoon and coastal Atlantic bottlenose dolphins at an important immune response gene. And then finally, my project was differentiation using multiple immune loci at high throughput sequencing. And this process can now be easily applied to additional populations and species. With that, I would like to thank everybody here at Harbor Branch who made this research possible, as well as other organizations and the funding from the license plate that allowed us to do this research. And thank you all for your time.